here and we're ready to go here. All right, so I promised you guys that I was going to put up that self-study video with the self-study slides. And again, I underestimated how long it takes to make those videos, and I finished making the video, but I did not finish editing it, so eventually it will come up. So just in case it worked the way I wanted it to, what would happen here is that, okay, first describing what is happening. You have two beakers. Okay, you have two beakers. In one beaker, you place a metal that is an electrode, that is a cation. Okay, so that's positive charge electrode. In the other beaker, you put another charged electrode, but a negative charge one, so that is an anion. Okay, so here you have electrode that is a cation, and here you have an electrode that is an anion. And when you run a current between these two electrodes, so here you have some current going around. Okay, current goes that way. So what happens here is that the water is going to start to communicate between the two beakers because of closing up of the current. Okay, and you're going to see that between these two beakers you're going to start forming a bridge of water. And this bridge is quite strong that when you take the beakers apart, this little bridge really stretches out up to about 4 centimeters or so. And when you look at it, it doesn't really look like it's liquid. It looks like it's kind of a gel. Okay? And I really like the movie, but for some reason it didn't work. Okay, so what is happening here? So what is happening here? Well, those simple experiments with water are really telling us that we don't quite understand how water behaves with each other, with each other's molecules, meaning here like the social behavior of water. So that's why I have this picture here taken from Dr. Pollack's class of, water molecules sitting in a bar and kind of like interacting with each other socially. We don't really know the nature of that behavior. And we also don't quite understand how molecules of water move around in relation to each other and in relation to other molecules. But we're really trying to understand more and more lately. Okay, so what is happening here? What was found in recent years was again looking here at interfaces of some um, substance and water. So here you have a gel, for example, if you are using like a polyacrylamide gel, that some people that have done DNA gel or protein gel electrophoresis, you're familiar with this type of colloid substance, right? So if you put a gel into this beaker of water, and then you put some particles dissolved in the water or in solution, puts particles in suspension in the water, like little pieces of black pepper, okay, in the water, mix in the water. So when you put this gel and you're going to have an interface between the water and the gel, just because the gel is there, what you observe is that this part that is close to the gel, in this area, you do not find particles. You see that there is this area in which it becomes totally clear of particles. So this area here that is close to the gel, this area is called exclusion zone. And it's called exclusion because the particles are excluded from that area. Okay? So this happens naturally. You don't have to do anything for this to happen. All right? It's just you put something in the water that is hydrophilic, and then particles are pushed away from that area, but the water molecules are still there. So what's happening here? Okay, that's the question. What's going on here? So we see that after you put the gel, the hydrophilic substance here, the particles kind of run away from that surface. Okay. So now when you start looking at what is happening with the water molecules that are closest to the surface, that is this hydrophilic material, okay, you find that the water is not really liquidy, so it kind of has this gel-like property. So when you look at the molecular arrangement of these gel-like properties, the molecules are not quite arranged like we expect in liquid water to be H2O. They kind of form these honeycomb shaped structures. And here we see the 2D representation, but if you think in 3D, you see that those really form like honeycomb shaped. Now, when you compare this to solid state water, like for example, if you compare this to a snowflake, you'll find that snowflake has those beautiful patterns that form those lattices. So we are kind of, you know, familiar with that, and we totally accept that as something that just happens, right? But this is not a snowflake. This is not solid water. This is something that forms these structures, that is a state that is between the liquid and the solid. So this is what we call the fourth state or fourth phase of water, is this gel-like structure, okay? And still, when you look at the molecular structure at this stage, you can't really say that it is H2O. Because these molecules of water are forming the lattices, you can say that this molecule is more like H3O2. So H3O2 would be kind of like this part of the molecule. And then when you look in 3D state, this would make more sense. But now you start thinking, oh, really, water is not H2O. Oh, we're talking H3O2. This is kind of weird. But then this is not really liquidy. This is more like a gel. Okay, so that's something different. So this area in which you have this exclusion zone, this is where you get these lattices. And then it gets even more interesting, because when you look at this property of easy water, now you can say that a lot of properties of water that we are used to, they actually start to make sense. For example, when you look here at those droplets that did not instantly get absorbed in the body of water, and you start thinking about, oh, look what is happening here. This floating droplet, when it is going through the air, right, between the beaker and the body of water, it is surrounded by air molecules. So this interface between the air and the water droplet, that allows the formation of the exclusion zone, okay? And then something else interesting happens, because at this exclusion zone, you start to get charge separation, okay? You start to get a lot of negative charges. They get very close to the water, to the little droplet of water. So here is separation when you're having a lot of negative charge. So farther from the negative charge, you start having positive charges. Okay. So now when you put two droplets, like not, of course, not stuck together, but side by side, like for example, when you see droplets of water that has evaporated, right, it's going to make a cloud. If you look at the droplets like microscopically, you're going to see that you have this exclusion zone formation. Close to the droplet, you have a lot of negative charge. A little bit away from the droplet, you have more positive charges. So when you put two droplets together, you're going to have an area that is negative charge, positive charge, and negative charge. Now remember, opposite charges attract. So what ended up happening is that these two droplets, when they are floating in the air, they end up getting close to each other. And that makes sense when you start looking at the macro organization of these droplets. Now it makes sense that clouds tend to be this shape of water stuff that gets close to each other. You don't see usually clouds that are just dispersed in the sky, right? You see clouds that make this type of big shape of water molecules close together. Okay, so this attraction between the water molecules is really coming about because of this separation of charge. Okay, so now you start looking at the strong properties that are adhesion and cohesion due to the interface of water and something hydrophilic, and this extra property that is the formation of this colloid state of water, these colloid crystals of water. Okay. So that's your easy water. And then it starts to get even better. I mean, it gets more interesting because next thing we should ask is this information useful? Okay. And how do you really can you make more easy water, right? And we just talked about charge separation. And we know that when you separate charges and you connect them by a wire, you end up getting a current. So this thing can really be a naturally occurring battery. Okay, so who in the class took chemistry, basic chemistry? Yay, one person. One person? Two, three. I hope most of you took chemistry. Just in case you didn't, I do have that review for basic, chemical properties of molecules and everything, and the movie will come up tonight because I'm finishing on the editing part. Okay, so if you need to review some of those chemical properties of molecules, just watch the video. I'm not going to be asking you a lot of details on that on the exam for this class for Unit 1, but you should understand how things are happening there, because then the rest of the class will make a lot more sense. Okay. All right. So talking here about the naturally occurring battery. Okay. Next thing we should be thinking is, okay, we have separation of charge and we have this kind of a battery going on here. I didn't turn on myself. So the question is, can we use it? Can we use this charge separation and this battery to as a battery to power up something? And the answer is yes, you actually can. Okay, if you put tiny electrodes in the easy water and farther away from the easy water where you have more concentration of positive charges, you can actually use this thing as a free natural source of electricity. And interestingly, the people who started to have these ideas, or one of the scientists who started to have this idea, um, was a person who should be very famous and is lately being referred to more than in past times. Anybody heard of Nikola Tesla? All right, who heard of Tesla? One person, two people. Who heard about that? Uh, notorious fight between Edison and Tesla. Anybody watch documentaries? Kind of interesting, right? Did you hear about what happened to Tesla's laboratory when he was trying to do exactly this, to harness um, electricity from the atmosphere, from water, and he really wanted to make electricity, freely available to everybody. So he was really working on that. You know what happened to this lab?
We have a guess up here in the front.
Yeah?
What did you say? It burned down?
Yeah, it got burned. It got burned. Some electrical companies really did not like that idea because they wouldn't really make a lot of money if electricity is freely available, just because you could harvest electricity from the atmosphere, from water droplets in the atmosphere. So yeah, his lab all got burned up. His notebooks got I don't know, disappeared somewhere. But the guy was really smart, right? A lot of the ideas that he had were actually used, recently with the development of those electric cars and things of that sort, right? So yeah, this idea is not new, right? It would be nicer if we would understand more about it, because biology and physiology would make a lot more sense, right? All right, so let's look a little bit more into what's happening here. These movies are giving me a hard time. But you can imagine here that when they turn the light on in the microscope, the water started to move through this straw spontaneously. Okay, so this is a movement of water that started to go through this tube. Okay, just because the light was turned on. So nothing else was done. There was no electrical charge. There was no electrode, anode, cation. There's nothing. Okay. The only thing that was done here, this thing was in a beaker or in a plate. The glass straw was in there. The light of the microscope was turned on, and the water started to move through the straw. And that is very, very significant, because now I started thinking, which other vessels are using water-based solutions or suspensions and also make use of movement of that suspension through tubes. And of course, I'm talking about living bodies, okay? Because we have all this cardiovascular system throughout the body, and we know that those are very long tubes, and they're filled up with suspension, that is the blood. The blood is a suspension because it has all the cells and all the stuff diluted or floating around in the blood, right? And those are all happening, all this movement of blood is happening inside of vessels that can be representative of phase separations, okay? So now the question is that if light can make this water move through a tube, is it possible that light can also make our blood move through our blood vessels in an easier way? Amazingly, the answer is yes. And what kind of light can make the circulation of blood easier on blood vessels? It's actually infrared light. Okay, so infrared light is making this thing, this liquid, to move through this interface and also within vessels, okay, within tubes. Okay, and a question: where do you find infrared light? What kind of bodies produce infrared light?
You don't have any guesses?
No guesses. All right, did anybody hear about night vision goggles?
Yes, people have heard that, yes.
Oh yeah, oh good. And what is it that the night vision goggles are detecting? Live bodies?
Some people have said heat.
Heat. And where is the heat coming from?
The body.
The body, like atoms, right? So if you put night goggles on and you turn off the light in the auditorium and you start moving your head around, what you gonna see?
Light.
Oh, you're gonna see everybody who is alive is gonna start glowing, right? Because you look at everybody that is alive, you have this whole system going on that is maintaining your life, and you are using energy, and your heart is pumping all this blood through the blood vessels. So the movement of blood and the beating of the heart is causing an electrical current to be forming around you. Okay, so you have an electrical current that is flowing around your body. You are producing infrared light just because you are alive. So that IR light is also helping your blood to circulate better. And now you start thinking like, oh, is it only infrared light? Well, actually, no. There is a lot of light that can make this circulation happen to a better extent. Like, for example, you go out in the sun, you have all sorts of radiation waves coming from the sun. And that is also helping the water in your body move around. Okay, so when you start thinking about the physiology, what goes on in the body, first thing that you think it come to your mind is we're all based in water, right? A lot of our bodies are really made up of water. And this property alone of easy water formation and charge separation is very much basic to the functioning of our cells and of the whole body. Okay. So with that, we finished the talk about the easy water. And if you want to hear more about it, follow the link for Dr. Pollack's, what do you call that? Those TEDx lectures. Okay. So now we're going to brush quickly through the idea of acids and bases. Again, this again together with charge separation, and here we are going to briefly review what is happening with this idea of acids and bases. Okay, so we know that acid is a proton donor and a base is a proton acceptor. Okay, and depending on how much of those hydrogen ions or just protons that we call them, right? They are in a solution in the blood, for example. We can calculate the pH, and our blood really has to be at a very narrow range of pH in order for us to maintain homeostasis and maintain life. Okay, so the pH of our blood really needs to be between 7 point, I believe it's 37 and 7.45 is a very, very narrow range. It's a little bit to the basic side, okay? But it's very, very close to neutral. And why is it that the pH has to be so controlled? Well, basically, we know that our life and cells are going to be dependent on the function of enzymes and proteins. So the rate of those proteins, the rate of those reactions are very much dependent on maintenance of optimal pH range. Okay. Besides the function of proteins, we're going to look at function of membranes and things that go in and out of membranes. That's called permeability of membranes. That is very much linked to proper pH range. So rate of enzymes, stability and conformation of the enzymes and of other proteins, and also the rate of the transport that is connected to the permeability of membranes, that is very much governed by changes in current or changes in charges. Okay, so pH, again, very, very important. Okay, as well as everything else that happens in the cell. So the cell, the liquid that bathes the cells, that has to be, pH of that has to be very much controlled. Okay. So here, acids and bases. If you have high concentration of hydrogen ions or protons, then you have an acidic solution, but your pH is low, is less than 7. So acidic pHs are going to range from 0 to 6.99. 7 is the neutral pH when you have equal amounts of hydrogen and hydroxide ions. And then when you have more hydroxide than hydrogen ions, you start to have more alkaline solutions. So more alkaline solutions means that you have low pH, low hydrogen ion concentration. That means high pH. Make sure you review these terms and you understand what is happening here. Okay? Low pH means acidic solution. High pH means basic or alkaline solution. And that takes us to what is happening with maintenance of the pH. And of course, we have to know a little bit about buffers, right? So what is a buffer? Well, a buffer can be a weak acid and its conjugate base, or it can be a weak base and its conjugate acid, okay? So a weak base, conjugate acid, conjugate base, okay, conjugate base, conjugate acid. All right, so what is happening here? How is it that buffers work? So let's see here. If you have a solution that has a weak base or a weak acid, okay? Let's say, for example, here we have a weak acid. You put in a solution, and then you have something that is going to steal hydrogen from that solution. So if you drop a strong base that steals a hydrogen from the solution of water, okay, what is going to happen is that if you do not have a weak acid, then that hydrogen is going to be taken away, and you are going to end up with very little hydrogen in your water, and that is going to mean that your pH is going to increase. Okay, so if you drop a strong base in the water and you steal that hydrogen, then you should have alkaline solution to end up with, right? But if you have a weak acid present that is working as a buffer, so this is not happening. Okay, so what is happening here is that when you drop a strong base in water and the strong base is still zero hydrogen, so now your weak acid is going to come into action and is going to donate a hydrogen to substitute that hydrogen that was stolen by your strong base. So now you're going to end up with a weak base, and you're going to end up with no change in your hydronium concentration because this one came from your weak acid. So this is the whole principle of buffering, okay? And I do hope that this is a quick review for basic chemistry, okay? So your weak acid here is substituting that hydronium ion and is maintaining the pH of your solution. So that was the base of a buffering, right? Buffering system. When you look in the blood, you're going to see that the blood has a couple of very important buffers, and the most important one is the carbonic acid bicarbonate system. Okay, so how is that working here? We know that we have here this is the carbonic acid, okay, carbonic acid that can dissociate into bicarbonate and proton, okay. So what happens here is that this reaction can go either way, okay, can go either this way or that way. This is why this is a weak acid and is a buffer, okay. So what's going to happen here? If you have in solution something that is going to steal the proton, so then this reaction is going to go forward, okay, it's going to go to the right here. If you have, to the left, depends on how you look at it, okay, right, left. Okay. If you have something that is donating protons, this reaction is going to go backwards, okay. So this is a very important buffering system for the blood because it's going to maintain the total amount of protons stable. So let's see here. This I took from another video that is very explanatory on this bicarbonate system. Okay, so let's see what happens here. First thing that happens is, well, how are you going to get carbonic acid in the first place? And which one how do you distinguish carbonic acid from bicarbonate? Carbonic acid is going to have one extra hydrogen right there. So, okay, so if you have a hydrogen to donate, that is your acid that is a hydrogen donor. Okay, so this is your carbonic acid. So this other one here with the negative charge, that's your conjugate base, or this is your bicarbonate. So what is happening here? When you look at a solution, okay, so water solution here, and you have carbon dioxide that is present in the water, you have the conversion of this carbon dioxide and water to this weak acid that is carbonic acid, okay? There is a very important enzyme that can do its conversion. So this is the catalyst for the conversion. This enzyme is carbonic anhydrase, right? So this is a very fast enzyme capable of lowering the activation energy of this reaction, right? So what is happening here? Carbon dioxide in the water where are you getting the carbon dioxide from? All right, let's think. One brave soul. In your body. You look in the blood.
That up front here. Go ahead. Byproduct of cellular respiration.
Yes. So just because your cells are alive, that's a byproduct. So you're producing carbon dioxide, right? So carbon dioxide is a gas. It'll diffuse out of your cells and it's going to go into the interstitial fluid. Eventually, it's going to get to go through the capillaries and it's going to reach the blood. So carbon dioxide is going to react with water and you're going to get carbonic acid because this enzyme is present in a few places. Okay. So now, carbon dioxide, when it's serving as a buffering system, it can donate here a hydrogen, right? And the other way around too. When you have too much acid, this can go the other way. Let's see here, the next slide. Okay. When you have, okay, first case here is too little acid. Okay. When you remove your hydrogen from the solution, when you take it away, so what is happening here? What is happening here is that you're taking away one of the products of this reaction. So your reaction is pushed forward. Okay, so you see this arrow going that way over here. So if you take out the acid, so your carbonic acid is going to give you bicarbonate in order to maintain the pH of the blood. Okay, so acid removed, and then you have this this state of maintaining the blood pH. When the acid gets removed, you get a state of alkalinity, right? Low acid concentration, that is alkaline, so alkaline tendency here. But then this is really good the other way around. When you have some extra acid that is being dumped in the system, so what is happening here is that you are pushing, based on the Chatoyer's principle, you're pushing this reaction that way, you're pushing it backwards. And then carbonic anhydrase is really going to catalyze the opposite reaction, right? And you're going to end up with carbon dioxide and water. So let's think about a situation in which this is happening in the blood. When do you think your body or your muscles, when do you think your muscles are going to produce more acid?
Someone said when you sweat.
When you sweat, why would you be sweating?
When you work out.
Yes, basically when you work out. So you're flexing those muscles. You are at the gym. You're really pushing hard, right? You're getting warmer. Yeah, you're sweating too. But basically, when you have more acid that's being produced by muscles, one example is lactic acid, right? Then you're dumping this acid in your interstitial fluid. Then this reaction is going that way, and you are going to end up with more carbon dioxide that you have to get rid of. So this is really cool because how do you get rid of carbon dioxide?
Exhale.
Yeah, you breathe. You breathe faster. Does that happen when you exercise? I hope so, right? You're exercising, you start to breathe faster. One reason is because you have to get rid of carbon dioxide. Another reason is that your muscles need more oxygen, right? But one reason is because you get rid of carbon dioxide. And this makes total sense because you're dumping more acid in your blood. This reaction is going backwards this way. This thing is becoming carbon dioxide. And yeah, it makes sense to get rid of it. Okay? So acidity in the blood will push this reaction backwards. So here is just a little diagram showing a state of homeostasis. All right, so this is a balance showing that you are in normal blood pH. So if you tilt this scale to one side or the other side, all right, so let's say here, when you have not a lot of hydroxide, of hydronium ions, right, when the arrow is going down here, okay? when your hydrogen ions go down, your blood goes toward alkalemia, okay, alkalemia, and that means it becomes more basic. I like this gnome with the base. You get a lot of bases right here. So you're tipping the scale towards the basic side or towards alkalemia, right? So in order to counteract that, you look at your bicarbonate system and you see which way the reaction is going to go. And here's the other pH tipping, right? When your pH goes more towards acidemia, the acids are always represented as red, right? So more acidemia, that means that your bicarbonate system has to adjust in order to bring your pH, to maintain your pH at a proper level so that life can keep on, going on. Okay. Is that more or less clear to everyone what's going on here?
We've got nods.
All right, good. So here comes your first question. Let's see how that's going to work. Ray, can you select the screen and start the iClicker? And hopefully everybody will be able to vote, to put in their answers. So the question is, a buffer will release hydrogen ions, right? If the blood pH, what? When do you need hydrogen ions to be released from that buffer system?
Five seconds.
Oh, is it counting down or is it counting up?
It was counting down. Okay.
Okay, so did we get
People are complaining even though the timer was on there the entire time. So should I open it again or?
Yeah, let me see. How many people need to answer it?
Like a quarter of the class.
All right, so it started again, but only the people who did not answer. Okay, only do not double dip into this question if you already answered it.
Okay, so if you've already answered, don't do it again, but we're gonna do another 30 seconds.
All right. And I will detect if you double dip into this question. So I'm glad that it worked, right?
I've got it all hooked up to my laptop this time with the desktop app and stuff, so.
Okay.
I'm just not even involving the computer in the room anymore.
Oh, really?
Yeah, it's too frustrating. Okay.
Okay. All right, so we got the answers there. What is the right answer?
People are saying A.
Yeah, so when your blood pH gets more basic, right? When the blood pH raises, this is when you have to dump more hydrogen. So your correct answer is A. All right, so the rest of the chapter, you can do the self-study video, and we're going to start talking here about the organic molecules, and I'm going to fly through this because, again, this should be common knowledge. You have 4 classes of these main organic molecules. One of them is carbohydrates. Other one is lipids or fats. Then you have proteins that are built up from amino acids. And lastly, you have nucleotides, and those are going to build up the what? Nucleic acids and genetic stuff. So let's see here types of reactions that are going to happen and involve these compounds. So basically, the building blocks are going to get together and form polymers, all right? The polymers are formed from monomers. Those are your building blocks, okay? And the way that you put those building blocks together, those are by reactions. There are condensation reactions, and they're also called dehydration reactions. Okay, so condensation or dehydration, those are terms that are interchanged. So what is happening here? If you have one monomer, get close to another monomer, and you always need an enzyme for that. You always need a catalyst for that. So you get them together and you take a water molecule to make a covalent bond, and this is your typical dehydration synthesis reaction. Okay. Now, if you want to break a polymer or a piece of the polymer into its monomers or components, you're going to do the opposite type of reaction. You're going to put in a water molecule to break that bond. So that is called hydrolysis, right? Hydrolysis will give you the monomers. Okay. So here's an example of reaction that involves either building up or breaking down of some carbohydrates, right? So here you have a molecule, two molecules of different types of sugars, or could be same sugar. In this case, you have fructose and glucose that undergo dehydration reaction, and you form sucrose molecule. Okay, you form a compound that has two sugar molecules. And if you go the other way and you do um, hydrolysis, then you're gonna get individual monomers. And now you should be thinking, oh, does that sound like a good test question? Like what kind of reactions are really important to build up things or to build or to break down things in the body? And the answer yes, that sounds like some good test questions. So here we go for types of chains or polysaccharides that we can form in the body. They're really, really important. Then they undergo also this dehydration type of synthesis, right? So one of them is glycogen. So for our bodies, glycogen is what we build both in liver and in muscle, and that is a storage of energy, right? Is a short-term storage of fast storage of fast energy. Okay, so how is it that you're gonna get this type of polysaccharide? You're gonna do hydrolysis, I mean dehydration reaction, and you're gonna build this these little chains that can be branched or mainly branched, right? And what happens here, we see that those are built from these monomers of glucose. Okay, so glycogen is carbohydrate storage in very important for animals to store, especially in the liver and in the muscle, right? And those are possible because of dehydration reaction. So dehydration synthesis, that's the name of the game here. Then when you need to use up the energy, then you make this molecule to undergo hydrolysis. Here, briefly looking at fats or lipids, okay? So when you look at lipids, you're going to see a huge chain of hydrates of carbons, right? So hydrate of carbon, so this is a fatty acid. So fat is just carbon and hydrogen. You know, if you took organic chemistry, you know exactly what I'm talking about here. And this part here that can be uh, hydrogen donor, this part is your acid part, the fatty acid part of the molecule. All right. What is one thing that is common among all the lipids? Well, they do not like to solubilize in water. So they are insoluble in water, and they can be either solid or liquid. So if you have solid lipids, you call those fats. If you have liquid lipids, you call those oils. Okay. Again, how do you get those lipids to synthesize? You can get a joining molecule like glycerol, and you do the hydration reaction, and then you end up with like triglyceride. So three fatty acids stuck to glycerol is like triglyceride molecule. That's very important for long-term energy storage, besides also serving as insulation, like body heat insulation, and also protection in case of you know, for organ cushioning and protection from damage. So besides those lipids, they're very much insoluble in water. You also have this other type of very important lipids that are called phospholipids. Now, properties of these phospholipids is that they are amphipathic. That means they have one part that is water loving, and they have another part that is water hating. Okay, so the water loving part of the molecule is called the polar head, and then the non-polar part is where you have the carbohydrate tail. These phospholipids are very important for the formation of cell membrane structure. So here we have a picture of phospholipid structure. Okay, usually you have two non-polar tails and one big polar head. And interesting thing about the phospholipids is that when you put several of them in aqueous solution, they tend to interact, they tend to get together and naturally form a bilayer. So this is a natural formation of membrane structure. You don't need to have catalysts, you don't need anything else to push them to aggregate in this way. They will do this naturally, okay? And the reason why they form a bilayer is that they have two hydrophobic tails. If they would have only one hydrophobic tail, they would form another structure. But with two hydrophobic tails, they form a bilayer. So one side of that bilayer is going to interact with the water, and the other side also is going to interact with the water. But then the middle part of this bilayer is really where you get the lipids part interacting. Okay. So now we come to the third building blocks of compounds in the body. Okay, so we have the third type, which is of macromolecule, which is a protein. Okay, so we know that proteins will make most of the functioning parts of your cells of the body. Okay, so you think about 20 to 30% of the mass of each cell is made up of proteins, and you have a lot of types of proteins. You have some that are enzymes. You have other that are structural. You have some other proteins that are globular and they are floating around in the blood. And you have some other proteins that are fibrous. Okay, also part of structural proteins. Okay, so we're going to learn a lot more about the protein functions. Right? So how do you get proteins? Well, we use the building blocks of proteins, and those are amino acids, and you get them together by dehydration reaction. And when you put two amino acids together, you end up with a peptide bond. Okay. So let's see here more on proteins. Proteins are very, very important and have very many functions. So briefly brushing the functions of these proteins. One of them is to coordinate movement. Then you also have structural and strength, mechanical strength. You have proteins that serve as communication methods. So you have receptors and you have molecules to bind to the receptors. So communication among the cells. You have some proteins that are very important for defense against foreign invaders. So those are antibodies. They're also types of proteins. You have proteins that are going to control differentiation of cells as well as the growth of cells. And you also have proteins that are going to transport things or store things. And finally, you have a whole bunch of proteins that are catalysts. Okay. So examples of different types of proteins. Okay, so let's see here. You have here your structural proteins. One example is collagen, and that is part of many tissues in your body. One of them that comes to mind right away is on your skin, right? So collagen is structural protein. You have other types of proteins that are enzymes, right? So those would be mainly globular proteins. And you have also transporters. So in this case would be hemoglobin. So those are also globular, and they're transporting oxygen and a portion of carbon dioxide. Okay. You have proteins that contract, so that's important for your muscle fibers, their muscles to contract. You have other proteins that help with communication. So those would be proteins on the membranes, and they can either be going across the membrane, or they can be on one part and communicate to another protein that is on the other part. So those are helping with messengers, okay? So communication. And here another example, that is your antibody protein for defense. So again, here looking at how is it that you get peptide or fusion of your building blocks of proteins. So we have here amino acids, right? And now you do not have to memorize all different types of amino acids, because that's biochemistry class. So this is not biochemistry class, right? But you should know basically what makes up an amino acid. You have an amino group with a nitrogen right there on one side, and you have a carboxylic acid on the other side. So this is your amino acid with a carbon in the middle. That's your alpha carbon. And you have a side group connected to your alpha carbon, right? So you get here an amino acid, you put close to another amino acid, and you have an enzyme to do that dehydration reaction. What is the name of that enzyme that will do this type of reaction to make a peptide bond? Does anybody remember? It's actually not quite a protein that does that. It's something else. Everybody still awake? Yes. Shout it out loud so then Ray can say what you're saying.
She was saying she was awake.
Oh, good. So what's the name of the enzyme? It starts with an R. What makes proteins?
Good.
Huh?
He said ribosomes.
Yes. Who said that? Okay.
Raise your hand up.
Okay, I see you. I see you. Hi. Yes, the ribosomes. And they are made of RNA, right? We do have some proteins in there, but the RNA itself makes this very important dehydration reaction. Yeah, but we're talking about proteins here, but then this is like making up of the protein, right? So catalytic reaction, dehydration, actually ribosome does that, but you end up with a peptide bond, okay? So peptide between carboxylic and amino group. You gotta know what a peptide bond is and how to get it. Does that sound like a good test question? Yes, so I gave you some test questions already. Okay. All right, so looking at some fibrous and globular proteins, and I'm going to take less than 10 minutes to go through this, whatever slides I can go through, okay? So we know that some proteins that are intended for structural purposes, they are actually kind of tough, and they are not soluble in water, which makes sense, because if they dissolve in water, they're not going to be very good to maintain the structure of the body, because the whole body is in this water base situation, right? So examples of structural proteins. You have keratin, that's for nails and hair and skin also, right? Elastin, collagen, okay? So collagen is really the most abundant protein in the body, okay? It's like everywhere, and different types of collagen too. Okay, so when you compare here your structural protein to something else that is globular, you see the structural proteins have all these types of fibers that a lot of times they get together and form helices for structural strength. And here I put an example of a problem that can happen with a genetic defect in only one type of collagen in the body. Okay, so this collagen is type 7, okay, and you do have 9 or more types of collagens in the body. And when you look at the different classification, you have the toughest ones are collagen type 1. Okay, so that you're gonna see later is found in tendons and ligaments, tough types of collagen. But then it gets softer, not as strong. And when you look at collagen type 7, this collagen is very important for adhesion of the skin to the underneath tissue, okay, to the connective tissue. When this collagen is messed up, your skin does not adhere to the connective tissue underneath. That means the skin, it naturally, it will just peel off, and the person can end up with lesions throughout the body, like most of the time, because the skin doesn't stay in place. And this is comparable to burns. So you can imagine this is a very, very painful condition. Okay, the name of this condition is epidermolysis bullosa. Anybody heard of epidermolysis bullosa, EB, in the class? Who has seen something like this before? Show of hands.
Yeah, some hands.
Some hands are up. Yeah, it's very, very sad situation. And actually, this boy is known from like my family knows him, and he's in his early 20s right now. He is still alive, but it's a very painful life. Okay. So you can follow the link and find out more about this. And if you imagine, this is one de novo mutation of collagen, right? So it happened in utero just by chance, and this boy ended up with this condition. And you can imagine also that his skin is very sensitive to any type of touch. If you touch him, the skin will peel off and you'll get blisters. So yeah, it's very sad. So those are your fibrous proteins or structural proteins. So here briefly going also through the other types of globular proteins. They are also making a huge percentage of the functional proteins we have, because they can make all different types of functional proteins like hormones, antibodies, and enzymes, and they're very diverse. Here is an example when something goes wrong with a globular protein, that in this case is hemoglobin, right? So a genetic mutation in the gene that makes the hemoglobin protein. If you have a change of one of the coding regions, for example here for valine, and the final product is mutated to a different amino acid, right? So you're going to get a defective beta globin molecule. And what is going to happen is that when your hemoglobin is saturated with oxygen, it looks normal, okay? But when it gives up the oxygen at the tissue level, then this hemoglobin, it gets a strange shape. Instead of just looking normal, like a little donut without the hole, right? Instead of looking like that, the hemoglobin starts to get this bent shape, which is called sickle cell shape. And when those sickle cells form at the tissue level, like at the level of capillaries, they are very, very fine blood vessels, that this, of course, they just deliver oxygen to the tissues. When that happens, these sickle cells, they tend to bump into each other, and they tend to form polymers, and those polymers clog up the very fine capillaries, and the person ends up with a lot of problem, because the tissue cannot be properly oxygenated, and you end up with some kind of plugs basically in those very fine capillaries. So this is also a very painful condition. Okay, so sickle cell anemia is a defect in this globular protein that is hemoglobin, and is caused by a genetic mutation. It was just one amino acid change that caused all this trouble. All right, so that also sounds like a good test question, right? Defect in a globular protein, hemoglobin, everybody should know that because I'm only giving two examples so far. Okay. All right, so briefly looking here at enzymatic activity. What is it that enzymes do? Well, they help a favorable reaction to take place. So what does that mean? So let's say here that you have, oh, that does not look too straight, that you have, and this is not going to work, okay, let's say that you have here products on one side and reactants on one side and then products at the other side. So this delta G is going to be spontaneous, so this is a negative delta G. Now let's say that in order for your reaction to happen, you would need to have a huge activation energy for this thing to get in proper orientation for you to go into product. This huge activation energy might make the reaction not very probable. But then here comes your enzyme, and the enzyme will put these reactants in proper configuration for the reaction to take place. So the enzyme lowers the activation energy and make the reaction more probable, right? So lowering the activation energy, that's the job of catalysts, and that's the job of the enzymes. So here's the picture I was showing you on lowering the activation energy. But again, your overall delta G has to be negative, otherwise your reaction is not going to happen. Doesn't matter how many catalysts you put in there. So here's a picture from the book showing how these proteins can do that job. They pick up the substrate or the reactants and they put it in a conformation that is going to make it highly probable for dehydration reaction to take place. Because most of reactions in the body, like building up anabolic reactions, happen by dehydration. Okay, so once you have your products, the products come out and your enzyme is recovered in its original shape. So again, another very important property of enzymes. They do not get consumed during the reaction. You start with this shape, you end up with that shape of enzyme, and that is a functioning enzyme. Oh, another question for you, just before we leave. So 1 second before you start it, Ray, let's just read the question and the options. So by the time you start that timer, everybody's ready to put in the answer. Okay. So when you start the timer, you guys wait about 2 seconds before you send in the answer. So that's the most effective way of getting these points. Okay. All right. So let's see here. Increasing the concentration of the enzyme's substrates. So those are the reactants. Okay, up to a point would do what to the reaction? So now you have to think about it. You have your enzyme with your catalytic side, and here you have your substrates that they have to actually get into the enzyme, to the catalytic side, okay? If you put a lot of substrates available, what is going to happen to your reaction? Is it going to slow down, speed up, or inhibit or destroy? Okay, so forget about those two. So it's really between slow down and speeding up. All right, Ray, let's start the timer.
All right, 30 seconds, guys. Can you see that it says 30 seconds in your app? No? Okay, sorry about that earlier. I thought you guys could see it.
Okay.
15 seconds. Two, one.
Happy New Year.
All right.
What
is the answer? B.
Yes, you're going to speed up the reaction. All right, so I see you all on Wednesday.
Yep,